Here's something that will change how you read your own DNA results. If you have ever taken a commercial ancestry test and seen the words Northern European or Western European in your results, those classifications were not calibrated against DNA collected across Europe. They were calibrated against a single reference panel built from 48 multi-generational Utah Mormon pioneer families. One community, one desert, one of the most genetically unusual populations in the United States. That community became the original global baseline that calibrated the entire commercial DNA industry, setting the foundational standard for what Northwestern European ancestry meant in testing pools worldwide. And to understand why that matters, to understand what that community actually is and how it got that way, you need to follow a trail that starts not with theology, but with ships, and ends with a cancer discovery that has saved millions of lives. The Mormon pioneer genome is not what most people assume it is. It is not generically British and Scandinavian. It is not a representative sample of Western European diversity. What it actually is, and why it ended up calibrating every ancestry test on Earth, is a story of accidental precision that nobody designed and nobody fully predicted. Start with the ships. Between 1849 and 1887, the LDS Church operated the Perpetual Emigrating Fund, which financed the emigration of approximately 90,000 European converts to Utah. The records of this migration, who left, from where, on which ship, in which group, survived nearly complete in church archives. The specificity is extraordinary. The pioneer genome draws disproportionately from West Yorkshire and Lancashire in England, from the Aarhus region of Denmark, from specific Swedish counties and specific Norwegian valleys. This was not a diverse cross-section of European immigration. It was a precisely documented sampling of particular regional populations, transplanted to a desert, separated from the rest of American demographic, mixing by geography and theology simultaneously. When commercial DNA companies later needed a documented, stable Western European reference population, nothing comparable existed anywhere else. The church had accidentally built the only one. I keep thinking about what it means that the genetic baseline for Northwestern European ancestry in every DNA test was set by a community whose founders walked across a continent to escape the world. The isolation was the point. The isolation was also, it turns out, exactly what made the genome scientifically useful. The migration built the genome. What happened to it once it arrived in Utah is the part that made it genuinely unusual. Between 20 and 30% of early Utah LDS families practiced plural marriage. The theological rationale involved building a righteous community and fulfilling Old Testament patterns. The genetic consequence was something population geneticists have very rarely documented in a modern Western population. The Y chromosome compresses when a small number of men father a very large proportion of the children in a community. Brigham Young fathered 56 children. Heber C. Kimball fathered 65. The Y chromosome haplogroups those men carried are now traceable in the descendants of those plural families at concentrations that reflect exactly this compression. The paternal diversity of the Utah pioneer community was narrowing toward the lineages of a handful of men across 3 generations of plural marriage. At the same time, the maternal diversity was moving in the opposite direction. The church's continuous recruitment of female converts from across Britain and Scandinavia meant that women carrying the mitochondrial lineages of dozens of different regional populations were arriving in Utah every year. The maternal genetic pool was expanding precisely as the paternal pool was contracting. The result is a population with one of the most extreme documented mismatches between paternal and maternal genetic diversity in any modern Western community. The fathers were few, the mothers were many, and the records of both, because of the theological requirement to document genealogy for proxy ordinance work, were kept with a precision that no other community in the world had matched. In 1892, things happened simultaneously that sealed this configuration in place. The church officially ended plural marriage under sustained federal pressure that included the threatened seizure of church property, and the American frontier closed. These two events together stopped the pioneer genome from receiving significant new genetic input from outside. What the rest of American DNA did over the following 30 years is the context that makes this significant. The Ellis Island immigration waves of 1880 to 1924 brought millions of Southern and Eastern Europeans into the American gene pool. Ashkenazi Jews, Italians, Greeks, Poles, Russians. The American genome became cosmopolitan. Utah's did not. The genetic distance between a fourth-generation Utah Mormon and a fourth-generation New York Italian whose great-grandparents came through Ellis Island is larger than the genetic distance between either of them and their respective European source populations. The Utah genome froze in 1890. The rest of America kept changing. What the frozen genome then revealed about one of the most central claims in LDS theology is a chapter that sits at the heart of the story. I find it harder to summarize cleanly than any other part of this evidence. The Book of Mormon presents itself as a historical record of a group of Israelites who left Jerusalem around 600 BC, sailed to the Americas, and became the ancestors of Native American peoples. This was not a peripheral church teaching. For most of the 20th century, it was foundational enough to the church's missionary relationship with Native American communities that specific programs were built around it. The Indian Student Placement Program placed Native American children in Mormon homes to help them reclaim what the church presented as their Israelite heritage. Native Americans were taught that they were Lamanites, descendants of Book of Mormon peoples whose history was documented in Scripture. In 2004, Simon Southerton, a geneticist and former LDS bishop, published Losing a Lost Tribe. The research was peer-reviewed and specific. Population genetic studies of Native American communities across the entire continent found zero evidence of ancient Middle Eastern DNA in any tested Native American lineage. The founding genetic signatures of Native American populations traced to migrations from Northeast Asia across the Bering Land Bridge approximately 15,000 to 20,000 years ago. There is no genetic signal of a Middle Eastern founding event anywhere in the Native American record. Not a trace. Not a marginal percentage. The data was unambiguous. The church's response evolved over the following decade. The official language in introductory material shifted from primary ancestors to among the ancestors. The framing changed. The programs restructured. But the specific people in Native American LDS communities who had been taught their entire lives that they were Lamanites, who had built their spiritual identity around a genetic claim that the science was now directly contradicting, received no official acknowledgement of what the DNA evidence meant for everything they had been told. I have thought about that silence more than almost any other detail in this research. The genome was doing something else simultaneously inside the pioneer community that took longer to fully recognize. When a population intermarries within itself for 80 years, which is what happened after 1890 when the frontier closed and the endogamy deepened, rare genetic variants concentrate. Variants that would remain statistically invisible in a large genetically diverse population become elevated in frequency within the isolated group. Conditions like familial adenomatous polyposis, a hereditary colon cancer syndrome, appear in Utah Mormon descendants at frequencies researchers can trace directly back to a single pioneer founder. Hereditary hemochromatosis traces through the HFE gene variant at elevated Utah frequencies, while specific cardiovascular vulnerabilities like long QT syndrome follow the exact same pattern. These are not random occurrences. They are the measurable biological consequence of the specific founding population, the specific marriage structure, and the specific isolation that the 1890 freeze extended for generations. The genome the church accidentally preserved is a medical record as much as a genealogical one. What that medical record eventually enabled is the part of the story that I was not prepared for when I started following the evidence, and it is the part that changes how everything before it reads. In 1974, a geneticist named Mark Skolnick walked into the LDS Family History Library in Salt Lake City with a specific hypothesis. The church's genealogical records were unlike anything available to medical researchers anywhere in the world. Pedigrees going 17 generations deep, nearly complete, cross-referenced with geographic and demographic information, maintained with the obsessive precision that the theological requirement for proxy ordinance work had imposed. Skolnick believed these records could be used to identify genetic risk factors for cancer in ways that were statistically impossible in less documented populations. He was right. Over the following 2 decades, Skolnick and his colleagues used the church's records to construct what became the Utah Population Database at the Huntsman Cancer Institute, linking 11 million individuals across 17 documented generations with modern medical records from the state of Utah. Researchers working with this database noticed that certain Utah pioneer family lines had elevated rates of breast and ovarian cancer across multiple generations. The genealogical records made it possible to identify all living individuals who shared those common pioneer ancestors, and to examine which of them had received the same diagnoses. The inheritance pattern that emerged pointed to a specific chromosomal region. Mary Claire King's laboratory identified BRCA1 in 1990. BRCA2 followed. The two genes responsible for hereditary breast and ovarian cancer, the genes that now form the basis of genetic screening programs worldwide that have informed the preventive medical decisions of millions of women across dozens of countries, that have driven the development of targeted cancer therapies that would not exist without the specific research pathway those genes opened, were identified specifically because they were elevated in frequency in the descendants of 19th century Utah Mormon pioneer families. The polygamy structure concentrated the variants. The 1890 freeze prevented those variants from being diluted. The endogamy that followed raised their frequency within a documented population. The documented population gave researchers the pedigree depth to detect an inheritance pattern that would have been statistically invisible in a more diverse community. The theology built the records. The records built the database. The database built the discovery. The discovery is still saving lives. There is one more thing the church built that is still operating. Under 700 feet of solid granite in Little Cottonwood Canyon, Utah, the LDS Church maintains the Granite Mountain Records Vault. The 14-ton doors were designed to survive a nuclear strike. The contents are 3 billion names on microfilm. The vault was built to preserve records for the dead, for the proxy baptism program that requires identifying specific ancestors across specific genealogical chains going back centuries. What it has become in the 21st century is something the people who built it could not have anticipated. The Family Search database, built from the vault's contents, now contains over 13 billion records spanning 6,000 years of documented human history. It is the largest genealogical database in the world. It is currently being integrated with commercial DNA testing platforms in ways that are producing genealogical discoveries at a scale no other institution can replicate. DNA tests that return results pointing towards specific surnames or specific regional origins can now be cross-referenced against genealogical records going back centuries. The vault that was built for the dead is generating discoveries for the living at a rate that no scientific institution designed specifically for that purpose has matched. The 3 billion names were collected to save souls. They are being used to find ancestors, to identify inherited disease risks, to connect families separated by migration and identity suppression, and to build the research foundation for medical genetics that extends far beyond the Mormon community itself. A community that walked 2,000 miles into the desert specifically to separate itself from the world, accidentally handed medicine the tools to save millions of people within it. Mark Skolnick walked into the Family History Library in 1974 and found records that took 20 years to fully understand and another 20 years to fully deploy. The genome that the polygamy structure concentrated and the 1890 freeze preserved became the statistically detectable signal that would otherwise have been invisible. The theological requirement to remember the dead produced a database. The database produced a discovery. And the discovery is still producing consequences. If this reached you, if you have Mormon pioneer ancestry and never fully understood what that genome actually contains, or if you have taken a DNA test and never thought to ask why your Northwestern European result was calibrated against a community in the Utah desert, or if the distance between what the Book of Mormon taught Native American families about their own ancestry and what the genome actually shows left you sitting with something that needed a name, drop it in the comments. Send this to someone who took a DNA test and never asked where the baseline came from. Subscribe if you want to keep following what these records contain. The vault in the mountain is still being microfilmed. The database is still expanding. Thanks for watching.